proteins can be attached to the phospholipid bilayer. There is a transmembrane proteins. These are proteins that span the entire phospholipid bilayer, and there can be one pass, three pass, or multiple pass. The second type in this diagram is called the monolayer associated, and you can see that it has a hydrophobic section of a protein going into the phospholipid bilayer, but not all the way through. So together, part A and part B are considered integral membrane proteins because the protein portion of it enters into the lipid bilayer. The third category here, called the lipid-linked proteins, are proteins that are covalently attached to a lipid, and the lipid is going into the phospholipid bilayer. And the last category here, typically I see it considered peripheral proteins, and these are proteins that are hanging on through hydrogen bonds or ionic bonds to another transmembrane protein. On this slide, the bottom diagram is showing the different types of functions that membrane proteins serve. So there are quite a few, and we'll cover some of them as we go through the different chapters. This picture is to show you that there is definitely an asymmetry in the membrane. So if you look on the outside, external side of the cell, you will see this layer of carbohydrates, and this is going into the extracellular matrix. All of the carbohydrates found on proteins or lipids are going to be on this side. On the cytoplasmic side, you would not see the carbohydrates at all. This slide is to show you that when we have these integral membrane proteins and they actually have the protein chain go through the phospholipid bilayer, that's called a transmembrane domain, and that domain is basically made of alpha helix secondary structure in the protein. That's about 20 hydrophobic amino acids in a row that extends the 4 nanometer across, which is the width of the cell membrane. When you have these transmembrane proteins, you can see that there has to be hydrophobic interactions between the alpha helix and the fatty acid chains. But right at the intersection of where the phosphate heads are, because the phosphate heads have a negative sign, negative charge, then there should also be a positive charge on the protein to allow the interaction through ionic bonds. And then, of course, there's hydrogen bonds that can occur elsewhere. In the biochemistry lab, if you wanted to study proteins that were found in membranes, one of the big issues is you have to be able to extract out these proteins. So the main thing is you need to first know, are you dealing with peripheral proteins or are you dealing with integral proteins? So if it's a peripheral protein, it's basically hanging on to the membrane through ionic bonds or hydrogen bonds to another protein. And so they're much easier, these kinds of peripheral proteins are much easier to solubilize because you can just change a buffer with pH or with salt conditions, and that will release those peripheral proteins. But if you're trying to work with the integral or the transmembrane proteins, now you have to deal with the phospholipids. So typically we use detergents that will solubilize the entire membrane, and then use other techniques to remove the proteins, remove the detergents from the proteins later. One of the main techniques that is used to separate mixtures of proteins is called SDS page. So polyacrylamide gel electrophoresis. We're actually going to do this in lab towards the end of the semester, but I'll just mention it here. When scientists were trying to figure out how many different types of proteins were found in the membrane, they again extracted the membrane, extracted the protein using detergent. And in this case, SDS is a detergent that has a very, very strong negative charge, and it will bind to the entire polypeptide chain and make it unfold and lose its three-dimensional shape. So basically, it denatures the protein into a single-strand polypeptide chain. The gel that is going to be made is kind of like Jello, and there will be holes put into the top so it can put in the samples. And so you put in the samples towards the top, and now that the proteins are all coated with negatively charged detergent, when you turn on the electricity, the molecules move towards the positive end, positive electrode. But because the gel is like a kitchen sponge, there's small holes and big holes. The smaller proteins will tend to move faster through this gel, and then larger ones will move slower. So at the very end, you end up with a protein pattern. So these little black lines show you the pattern in this case, and the smaller proteins are going to be bands at the bottom, and the larger proteins are at the top. One last technique, if you get proteins and you're able to sequence them, one of the things you can do is to put the sequence through a DNA program, computer program, and check for the hydropathy analysis. So the hydropathy analysis is basically a program to predict whether or not there are transmembrane domains. So remember that a transmembrane domain has about 20 hydrophobic amino acids in a row in order to span that liquid bilayer. And we have a program that will assign the R groups of every amino acid the number, depending on whether they're hydrophilic or hydrophobic. And when it gets a whole series of hydrophobic amino acids in a row, then it assigns it a higher and higher number. So look on the graph on the top here, and you can see all the numbers above 0 are hydrophobic R groups, and all the numbers below are negative numbers are the hydrophilic R groups. And what we want to do is we want to graph the R groups numbering based on this computer program, and if there's a peak that reaches past plus 1.8, then we know that it has a transmembrane domain. For the test, you will need to be able to analyze this hydropathy plot and then be able to draw a picture of the protein in the membrane, just like the picture at the bottom. And this picture is just to remind you that, again, the proteins found in membranes have many different types of jobs, and we will be